This protocol describes an alternate rapid approach for assaying insulin secretion from beta cell lines to expedite drug discovery and characterization. This approach is useful because multiple treatments can be tested simultaneously, the results are available immediately following the experiment, and this assay is more affordable than others. The application of this method towards screening for new small molecule modulators of insulin secretion may identify lead compounds for future diabetes therapies. Increased knowledge of beta cell function can contribute to greater understanding of protein or neurotransmitter secretion in general. This insulin secretion reporter has been established to work in a lentiviral system, in other beta cell lines, and in human islets. This assay should be very straightforward to individuals familiar with cell culture and operating multichannel pipettes. Before planning any experiments, verify that your parental or stable beta cell lines are responding properly to glucose using an insulin ELISA.
Begin by preparing Krebs-Ringer bicarbonate buffer, or KRBH, and Gaussia luciferase working solution according to the manuscript directions. Grow Min6 cells in T75 flasks using standard cell culture techniques. Prepare the InSG-Luc Min6 cells for plating by washing a confluent flask of cells twice with PBS and adding 2 milliliters of trypsin. Incubate the cells at 37 degrees Celsius for about 5 minutes, or until the cells dissociate from the flask. Determine the cell concentration and dilute the cells in complete media to 1 million cells per milliliter, which equates to 100,000 cells per 100 microliters in each well of a 96-well plate. The cells should